And also, like, this is it for the reviews. I mean, obviously, like, we've had Sydney do reviews. Now I'm doing an extra review. There's not going to be any more review, like, right before the exam or anything. So this will be it. We're going to get started with Unit 2 review. We'll go through, and if we generally get questions right, I'm not going to spend a lot of time on them. And if you want me to review something in more detail, just let me know. Okay, we're going to start with Chapter 6 information, which is our genetics chapter. First question from Chapter 6, which term refers to all existing versions of a gene within a given organism, even if they're not all expressed? So this kind of deals with the semantics and wording of some of our terms and the definitions associated with them. Here, in case I need it. All righty. I figured that it would be this way because it was for my Monday class as well. So genome doesn't refer to existing versions of a single gene. It refers to all of the genetic information in an organism. So that's why I kind of emphasized that when I was reading the question, right? So if we're talking about all versions of a single gene, remember that you get a gene from your mom and a gene from your dad. That's all versions of a type of gene. The gene for eye color, the gene for how tall you're going to be, the gene for hair color, and whatever. So genotype refers to all of the versions of a gene that are in an organism. And you might only express one of those versions. You might express a combination of those and whatever. But what we see physically or physiologically is going to be your phenotype. But the different genes that you actually, the different versions of the genes is known as the genotype. And then, of course, all of the genetic information in the organism is known as the genome. Right, so next. Okay, complementary strands in double-stranded DNA have phosphate sugar backbones running in the opposite orientation. What is the term for this? The backbones are all going in opposite directions. All righty, so the term for this is anti-parallel. One strand is running 5' to 3', and the other strand is running 3' to 5'. And they're therefore running in opposite directions. Remember that in genetics, we generally think of forward to backward, that would be 5' to 3', typically. Okay. So one strand running one way and the other strand running the other way. When we replicate our DNA, and we have one old strand used as a template to make the new strand, that's a process that we call semi-conservative, but it's a different concept than the anti-parallel concept. Okay, and you need to know the difference between those two. Hint, hint. Okay. All righty. In DNA replication, what is the term for the DNA strand that involves production of Okazaki fragments? If we're replicating DNA, and this only happens in DNA replication. We don't see this forming in RNA transcription or anything like that. Only in DNA replication. Alrighty, so this is the lagging strand. The strand that is made in fragments, because as we open the fork, then we can make another fragment. And then we open it further, we can make another fragment. Okay, and those fragments are called Okazaki fragments. And that happens on the lagging strand. The leading strand is made continuously. Okay. All right, moving on, because I'm pretty sure the next question relates to the other thing that people chose. Which enzyme seals the Okazaki fragments together after DNA replication? So if they're in fragments, we've got to join those fragments together. Nucleotides in DNA have to be joined together by a type of bond called a phosphodiester bond. So this enzyme is involved in rejoining, creating that phosphodiester bond between the Okazaki fragments, and that's DNA ligase. Okay, so that's where that one belongs. Good job. All right, beautiful. All right, DNA polymerase can only add nucleotides in one direction, and what direction is that? If we were paying attention. All right, so this is 5' to 3'. Well, we generally agreed that it should be one or the other. So we do add nucleotides to the 3' end, but the overall direction is 5' to 3'. Adding it on to that, extending in that direction in the 5' to 3' direction. Right. What is the process of making a working copy mRNA using DNA as the blueprint? DNA is our template, and now we're making an mRNA from it. What is that process called? This one, I wish I had music in the background that said don't like the quiet. All right, so this is the process called transcription. So let's go through kind of all of these because these are all important. We have to know what they all are. Replication is making copies of DNA to DNA. Transcription is making RNA from DNA. Translation is making protein from RNA. And then transduction is what happens whenever a virus inserts its genome and gives you new traits, resulting in lysogenic conversion. Okay. So that's what all those terms are. Are they all related to genetics? Yes. Okay. I'm sorry, but it just is what it is. So be sure that we are kind of knowing the difference between these terms. Yes, Megan.
You said translation.
Translation is using RNA to make protein. The way that I remember this, and I don't know if this is helpful for you guys, but I think of scribes as writing. So if I was a scribe and I was reading like our textbook, for example, I'd be making down my notes. But was the textbook in English? Yes. Are my notes in English? Yes. They're still in the same language, right? Just like DNA is nucleic acid and RNA is nucleic acid. So that's transcription, okay? And then translation is literally translating it into a different language. So we're taking nucleic acid language and then translating it into amino acid language. And so that's how I remember the difference between the two, because amino acids are a different language than nucleic acid. Proteins are made up of amino acids, for what it's worth. To a little bit more depth than that here in a little bit. All right, the mRNA codons are complementary to the anticodons on which molecule? So mRNA is made of codons. Where do we find anticodons? All right, if our mRNA is on the tRNA, so if our mRNA looks like this, and we have a codon here, and a codon here, and a codon here. Again, what is the codon really? AUG or CCZ or GA, who knows, right? So there's our mRNA codon that then the tRNA comes in and matches it up and brings in an amino acid. And then this right here is the anticodon. In this case, if that was AUG, this would be UAC, for example. Okay, so that's what the anticodon is. And this happens inside of a ribosome, kind of clamps down on all of this. And this happens inside of the ribosome. And the ribosome itself is made up of ribosomal RNA or rRNA. But it doesn't rely on matching up codons or anticodons. It's just the structure within which all of this would happen. Again, its shape and what it's made of helps bring in tRNAs and things like that. Okay. So the tRNA anticodon matches up the mRNA codon. And I think the next question is going to talk about what happens next. So this, what process is that I'm drawing out? This is translation. This is translation. Right. The building blocks needed to synthesize proteins that are carried by the tRNA. We actually learned in Unit 1 about what these building blocks were. It was in Unit 2 when we learned about the tRNA carries them. So those are the amino acids. Amino acids are the building blocks of the protein carried by the tRNA. And again, because the anticodon matches the codon, it knows to bring in whatever amino acid. For this class, you don't need to memorize your amino acids or codons or anything like that. I made that pretty clear early on, so you're welcome for that. But have an idea of what it all represents. So if AUG matches up with UAC on our anticodon, on our tRNA, then that tRNA knows that it's bringing in methionine, for example, as an amino acid. And so whatever that codon is tells the tRNA which amino acid to bring in. And that's how translation works. All right, next. Doing good. All right. What type of bacterial operon is always on, but will be turned off if the product isn't used fast enough? It's on and can be turned off. So this is a repressible operon, repressible. So we know this because if it's on and it can be turned off, it can be repressed, you know. Is the action of repression important in inducible operons? Yes, but they start with the repressor already bound, so their operon is already turned off, and then they're able to release it and then turn on the operon as a result of that. So, yes. Any questions about the operon stuff? It's a lot of people. You guys did pretty good on that question. All right, which type of mutation is occurring in this image here? So here we have one CAT got changed to CCT, and instead of coding for histine, we're now coding for proline. What type of mutation is that? Changed one nucleotide, which changed our outcome amino acid. All right, so let's see. So if in this process of translation, you know, we're telling through the codons, this is translation, through the codons which amino acid to bring in, right? And so in the picture that we saw here, we saw CAT turn into CCT. And instead of coding for histine, we're now coding for proline. So what this means is that if our original is CAT, and we're going to go through all of them. That's why I'm kind of writing this one down, okay? If CCT, and the C is our change, leads to proline, the original was supposed to be histidine, then this is what we call a missense mutation. If this had changed instead of CAT, let's imagine that instead it was UAT, I don't know what, but the one change, just one change still. And instead of coding for histidine, it's now a stop, then that would be a nonsense mutation. If we change CAT to CAC, again, just one nucleotide change, it doesn't matter whether it's the first or the second or the third nucleotide. So don't be focusing on that. If you're like, oh, it's the first one, that means it's no. What matters is what the outcome is. If it is a different amino acid, versus a stop, which is no amino acid, versus if CAC still codes for histidine, did we change the amino acid there? No, it's the same. This is called a silent mutation. No change in the amino acid. On exam questions, I will give you either a picture like this one, or I'll give you a story about that has the same amount of information in it, okay? So I'm not going to expect you to know that because you change CAT to CCT, that means you're going to have a different amino acid. I will tell you if it's a different amino acid. I will tell you if it's the same amino acid. I'll tell you if it's a stop, okay? And then you can tell me if it's a missense or a nonsense or a silent mutation here. All right, and so one of my other students also asked me in my other class about a frame shift mutation. And so a frame shift mutation would be where, like, you normally read it as CAT, 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 and whatever else comes afterwards, okay? Which normally codes for histidine, 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 and obviously that's the amino acids that that protein needs to have in that order. In a frame shift mutation, delete one or two nucleotides. Now suddenly that results in CAT, CAC, ATC, AT, whatever else came afterwards. Okay. Now you can see you have histidine here. I don't know what CAC codes for, ATC or whatever, but it's not the same thing. You always read in 3 nucleotides. So if you remove a nucleotide, you don't just skip a spot. You read the next one. This is like taking a book and deleting a letter and shifting all the letters downstream of it over by 1 letter, but keeping the same word sizes. And now you can't read it. It's not the same words anymore. So you can see how detrimental a frame shift mutation can be, as well as a early stop. Imagine having a stop at like your second amino acid where you're supposed to have hundreds of amino acids afterwards. And so these can be very detrimental and very deadly. And that can happen as a result of just 1 nucleotide changing. And the effect of 1 nucleotide causing potentially different outcomes, that's called a single nucleotide polymorphism. That was in, I think, chapter 8. So I just mean that there's different versions of outcomes based on 1 nucleotide difference. Any questions about this? I went over this because this was one that another class had a question over. Here are these drawings, good. Okay. So for chapter 6, know all the major terms for it, explain the steps of DNA replication, why Okazaki fragments exist, the leading strand, the lagging strand, understand, compare, and contrast transcription and translation, know the difference between the two, explaining operons and identifying types of mutations, and very big picture stuff for this. Know your learning objectives for chapter 6. All right, we're moving on to chapter 8 now. In DNA gel electrophoresis, which molecule will travel the farthest? Think of farthest as representing the fastest. Right, so this would be 100 base pairs because it is our smallest one. Because we have to keep in mind here 100 base pairs. This is 100,000 base pairs. This is 1,000, and this is 30,000. So just adding on those numbers on there. And again, when you guys do this, whether it's on the lecture exam or the lab exam, I recommend writing it out just so that you aren't going to get confused when you make your choices, because it's kind of easy just to look at the numbers and see, oh, this is a smaller number, but it's not. Because what does K mean? It means times 1,000. Okay, so kilo means 1,000. What does M mean if we had an M? Million. So keep that in mind too. It's easy for those Ks to get disappeared in there. So. Alrighty, what does PCR stand for? What do the letters stand for? All right, so PCR stands for polymerase chain reaction, and it's talking about DNA polymerase here. So it's causing a chain reaction resulting from the action of DNA polymerase. We're going to talk about it a little more. There's more questions about PCR, so we're going to feed into those questions next and then elaborate a little bit further. What is the purpose of PCR? The purpose of PCR is to replicate DNA. So if we were talking about creating DNA using protein as a template, which was a popular choice on this one, I'm not really sure how that would even work logistically. So right now, as it exists in all of modern molecular biology, we cannot create DNA from protein template. What that would mean is taking a protein and reverse engineering DNA out of that protein. That's what that would mean. And so based on that, so using protein as a template from which we would make DNA from doesn't exist in science, okay? And that we understand that because you have to go from DNA to make RNA to make protein, because protein's made up of amino acids. And these are obviously made up of nucleic acids. This process here is called transcription. And this process here is called translation. We have learned that you can actually go back from RNA to DNA. But this is a process called reverse transcription. Can your cells do that normally though? No. So you have to be HIV or something like that. They have a special enzyme called reverse transcriptase that some viruses have that allow for that to happen, but your cells can't do that naturally. So this is how biology works. DNA is the template from which we create RNA, which is a working copy, from which we will read and then know to match up our codons and anticodons and bring in the amino acids there. So if we were using protein as a template, using the amino acids to reverse engineer what your RNA or your DNA look like, is impossible because there's a lot of overlap on which codons might be there and stuff like that. So it's not possible to do that, okay? So PCR, so the way that it works is you start out with your DNA, one DNA, which then separates out its strands. Right. We will find a primer. Anytime. There we go. In the annealing step. And if the primer binds, then we can add on nucleotides. You can't add nucleotides to nothing. You have to add them on to the edge of something else. So this part here is extension step or elongate step, however you want to word it. If I ask you about the name of the step, you can even just describe the step and that would be good enough for me, okay? But this is where DNA polymerase adds the nucleotides. This is the same kind of process what we see with replication, okay? It's DNA replication, which is making copies of DNA, but it's just hijacking that. And again, it's depending on whether or not that primer anneals. This step of separating the strands uses high heat, and we call it denaturing. Because you can just break the hydrogen bonds that hold the strands together using just heat. Okay. So that's the overview of PCR. Any questions about that? Okay. One last thing. If we start with one piece of DNA, how many pieces do we end up after one round? Two, like this. One begets two. If we go through a second round, how many would we have? 4. 2 begets 4. And then after the next round, 8. And after the next round, 16. Good. So if I were to ask you after 3 rounds of PCR. This was after 1, this is after 2 rounds, this is after 3. After 3 rounds, we have for example, I might ask up to 6 rounds. You can draw it out. You can do a little math and double it out, or you can just remember. I don't care. I'm not saying I'm going to ask about 6. I might ask you about 3, 4, 5, 6. It's not going to be 1 or 2 probably though. But FYI. All right. Good. This, by the way, if you are a math person, if you're not a math person, shut your ears for a second. If you're a math person, the equation for this is 2 to the n, where n is the number of rounds. It's an exponential growth. All right, next question. In PCR, what is the purpose of the highest temperature step? All right, so it separates the DNA strands. Yes, so the highest temperature step, the hottest one, basically makes it so we're not having any hydrogen bonding. So it separates the hydrogen bonds, separating the DNA strands. We need to cool it down a little bit in order to bind our primers and extend along the sample. Okay. Good job. Next question. Where on target DNA do restriction endonucleases typically act? Restriction endonucleases. This is a different concept in chapter 8. All right, so restriction endonucleases. This was an issue with the other class as well. So this is reading and comprehending what the question is asking. It's asking where on the DNA do the restriction endonucleases act, not what do they create when they act. So when restriction endonucleases cut, they do create sticky ends, but they don't target sticky end DNA. They create the sticky ends. So that's kind of getting what the question is asking there. So 3' to 5'. So they act at palindromic sequences. So if I have a G here, what does G bind to? C, and A to T. Don't think too hard about it. G. Okay. GAATTC from 5' to 3' direction. What does this read from 5' to 3' direction? GAATTC. Is that the same? So that is a palindromic sequence. It reads the same on the forward strand as it does on the backward strand. And then your sticky end cutting would happen something like this and create the sticky end. So the restriction endonucleases cut at palindromic sequences and then create sticky ends. Does that make sense? I mean, you don't need to have to talk about the chemistry of it, but have a general idea of how that works. True or false? Restriction fragment length polymorphisms can be used to identify suspects in a murder. We kind of talked about this in lab. Yes. Okay. So it definitely can. Again, relating based on if I cut my genes at this palindrome, then I'm going to have different fragments that result than if I cut your genes. And we can run them out on a gel and see those different fragments. And that serves as a fingerprint to identify people. Those are restriction fragment length polymorphisms. We'll talk more detail about them in chapter 18 in unit 3. All right, what is the term for organisms whose DNA has been altered using genetic information from other animals? This could be even our E. coli that we transformed with our plasmid, with pico plasmid. We put jellyfish DNA into those E. coli. We didn't actually do it, but in theory we did, right? And so that would be a transgenic organism, also known as a genetically modified organism, or GMO. And so when we are actually literally taking DNA from another organism and putting it into another organism through molecular genetics, that creates transgenic or genetically modified organisms, GMOs. Alrighty, what is somatic gene therapy? This is what I would call the definition of it. This is the best representation from our choices. Alrighty, so somatic gene therapy has to deal with treatment that is not heritable. So heritable gene therapy would be called germline. So germline is heritable. And it's only achieved by manipulating an embryo or the fetus, in which case that could be inherited and passed down. Somatic gene therapy is affecting cells that already exist in the organism, not the gametes. So if we could affect gametes, which is sperm and egg, or if we can affect the embryo while it's developing, and therefore its sperm and egg are affected, then that would be heritable germline. Somatic is body cells. That's what literally somatic means. That's like body cells. And if we are causing gene therapy in existing body cells, the best comparison would be adult cell modification here, because the other ones would be germline modifications. All right. Same kind of review for this chapter. We're going to move on to chapter 9. What is the term for an organism that uses inorganic carbon as its carbon source? Alrighty, so this is getting back to all of our tropes and dealing with where are you getting your energy and where are you getting your carbon? What kind of trope are you? So if we're talking about where your energy is coming from, it can either come from the sun, which we call phototrophs, or it can come from chemical breakdown, what we call chemotrophs. Chemotrophs break down chemicals to get energy. That's what we do, right? Phototrophs get energy from the sun. Where do they get their carbon, though? These are autotrophs that get it from inorganic sources, or heterotrophs that get it from a variety of inorganic sources, typically made from, from organic sources, typically made by autotrophs. Okay, so the heterotrophs usually depend on the autotrophs. So if we were talking about human beings, they are chemoheterotrophs. We're talking about plants, they are photoautotrophs. We're talking about bacteria, they could be either one. They can also be something called lithotrophs. Lithotrophs get like inorganic carbon from like rocks and stuff like that. I'm not going to test you on that. You know, I'm going to test on it. You have to know these. Oh, okay. But there are other combinations. Okay. Moving on, what type of organism gets its energy by breaking chemical bonds in its food? Gets its energy from breaking chemical bonds in its food. Excellent, chemotrophs, chemotrophs. All right, which of the following terms best represents the temperature range for a typical human pathogen? These were terms from chapter 9. Let's just see if you guys can remember what term this would, what type of organism it's applied to. And then we'll talk about what each one of these mean. All righty, so psychrophiles like it cold. There is a specific temperature range. I'm not going to test you on the specific temperature range, okay? Mesophiles like mid temperatures. We like mid temperatures. Temperatures that you can survive at are mid temperatures, okay? So if it's colder than what you can survive at, that's psychrophiles, okay? If it's hotter than you can survive at, that's thermophiles. So thermophiles are organisms that like it hotter than what you typically would survive well at. We even have extreme thermophiles. Extreme thermophiles will flourish above boiling. It's crazy. But they actually prefer to be above boiling temperatures. Halophiles prefer salt. This has nothing to do with temperature. These are guys that exist well in salty environments, halophiles. Okay. A lot of our archaea fall into that category. Any questions about these terms? So mesophile, mid-temperature, temperatures that you would survive at. Meso means middle. All right, cool. If a cell is dropped into a super high salt concentration solution, what term best describes the salty environment regarding the cell in respect to the cell? Typical cell dropped in a high salt solution. What would you call the salt solution? This would be hypertonic. So what would happen to the cell as a result of that? It would shrink, right? The water would move out of the cell. Anybody need me to talk about that stuff? Hypertonic or hypotonic or isotonic? What's happening during the stationary phase of bacterial growth? Alrighty, so in our bacterial growth curve, we have lag, log, stationary, death. Lag, log, stationary, death. Okay, so in the stationary phase, what's happening is growth rate has slowed. Some cells might die just because they're old or will run out of a little bit of nutrients or whatever, but we'll have enough cells still be able to multiply that we keep it at a steady population rate. So that's the stationary phase. Okay. This part of preparing for growth and adjusting to the environment is the lag phase. The lag. Alrighty, which symbiotic relationship involves one organism that benefits, but the other organism neither benefits nor is harmed? Unaffected. Nose is tickly today. This is commensalism. Commensalism. All right, so for chapter 9, be aware of your terms for it. The important elements, chomps, right? So that's CHONPS in order of how common an element is in living beings. Carbon versus the energy sources, those were our tropes. Categories for temperature preference, those were our phials. Then we have diffusion and osmosis, the tonicities, and transport across the membrane, and the symbiosis, and the bacterial growth curve as well from chapter 9. Moving on to some chapter 25 questions. Select all that apply. What are the major terms for the major types of nitrogen cycles? We have a general overview of this. We don't need to know this in super specifics, but I just want to reiterate kind of a role of each in the nitrogen cycle. Okay. There's 3 major steps in the nitrogen cycle. If you have N2 in the air, it's the most common component of the atmosphere. So it's like 72% of our atmosphere is just N2, which is nitrogen gas. Converting N2 into NH3 or NH4 plus, this is a process called ammonification. This is done by certain types of bacteria, even in your gut, even in your body, okay? And then conversion of ammonia compounds into nitrite or nitrate, which are what NO2 is nitrite, NO3 is nitrate, okay? This is called nitrification. This can be done by some types of bacteria, and some plants can do this. Most plants can use nitrate and then convert it into amino acids, convert it into nucleic acids and stuff that can be used by the cell after that. So most plants can use nitrates there. And then conversion of the nitrate back into nitrogen gas, again, done by bacteria, is called denitrification. If you're wondering, like, okay, but where do I fall into this? We're not talking about you, we're talking about the microbes. But where you fall into this is whenever you eat protein and nucleic acids, that you will process out ammonia as waste. And then as ammonia, basically as ammonia or as urea, which gets processed down to ammonia, and you expel that in your urine. And again, that's ammonia that bacteria can turn into nitrates that plants can use, for what it's worth, okay? But you're not going to test it on that part, but you need to know that ammonification, denitrification, and nitrification are the steps that are involved in the nitrogen cycle, okay? Regarding microbes. Nitrogen detoxification is not a word, not a thing. Okay, 34, all right, in wastewater treatment, what is the term for the bacteria that act as indicators for more pathogenic organisms? If these guys are here, pathogens could be there too. Right, so these are fecal coliforms and normal poop bacteria. Yeah, if you straight up ate fecal coliforms, it would make you sick, okay? But in small numbers in water, it is safe for them to be released back into the environment and be watered down by the environment and stuff like that. But if normal poop bacteria can make it through the process, so can pathogens like cholera and salmonella and shigella and giardia and things like that. Not all poop bacteria are bad. Okay, so we're going to move on to chapter 10. What is the term for the protein part of a holoenzyme? Righty, it's called apoenzyme. So there's two types of enzymes that you can have. Simple, which is just protein, and conjugated. In a conjugated enzyme, you have a protein portion, which is called the apoenzyme, and a non-protein portion. This is called the cofactor. If the cofactor is organic, an organic molecule, meaning we've got carbon bound to hydrogen somewhere on it, then we would call that a coenzyme. The whole entire thing, protein plus cofactor, is called the holoenzyme. And this is for conjugated enzymes. So examples. Hemoglobin does not function without iron bound to it. The hemoglobin, the protein part of it, is called an apoenzyme. The iron would be considered a cofactor. Okay. If it's an organic molecule, like vitamin C is ascorbic acid, right? If you have an organic molecule, like most of our vitamins are, then they would be a coenzyme. But just because you're organic, does that make you a protein? No. Don't get confused. So, any questions about that? There's another question about it, I think, on this. True or false? Coenzymes include metal ions like iron or zinc. False, because a coenzyme, a coenzyme has to be an organic molecule. Is iron organic? No. Is zinc organic? No. Okay. Cofactors might include iron or zinc, but not coenzymes. Coenzymes have to be organic molecules. True or false? Competitive inhibition involves the product of a reaction binding at the allosteric or regulatory site on the apoenzyme. Was false. If this is normally how this works, the blue would be the substrate, right? And the purple would be the enzyme. Okay. If you have an inhibitor that binds where the substrate binds, an inhibitor that looks like the substrate but isn't quite the same thing, this site where the substrate binds, what's the name of that site? Active site. Is that the same word as allosteric or regulatory site? No, it's not. They're not the same. However, imagine you have your enzyme, you do take an inhibitor and it binds somewhere else, and then that causes your enzyme then to close up. Can't bind on the substrate because this thing bound somewhere else, caused the protein to change shape. This is called non-competitive inhibition. We're not competing for the active site. And the site where the non-competitive inhibitor binds is called the allosteric or regulatory site. Okay, so competitive inhibition, which is what the question is asking about, involves an inhibitor binding at the active site. Non-competitive inhibition involves an inhibitor binding at the allosteric or regulatory site. We've seen why this is false now. Anybody have any questions about this? All righty. Next question. If a molecule is reduced, what does that mean? I want to give it away. Okay. Leo says GER or oil rig. Whatever one works for you, but you need to lean on one of these. For my class, for physio, and if you take any more chemistry, some of you guys are going to take more chemistry. So Lose electrons and you are oxidized. Gain electrons and you are reduced. So if you're reduced, that means you gained an electron. Okay. Oxidation is loss or reduction is gained. That's what oil rig means. But either one, whatever works for you, but have some sense of that. All right. If a molecule is reduced, then it gained an electron. It accepted an electron. Okay. Anytime you hear about oxidation reactions or oxidation or oxidative phosphorylation, hint, hint, or something like that, it's talking about redox reactions always. And most reactions in some way usually are redox reactions. Something is gaining an electron and something is losing an electron. So having that reference in there. Okay. Next. Hydrolases, what do they do? Hydrolases hydrolyze. So that's what they do. What does that mean? So a hydrolase is an enzyme that breaks down large molecules into smaller ones. So amylase is a hydrolase. And what it does is it breaks down starch. So starch is a very large, complex carbohydrate that can break down, amylase can break down starch into sugar molecules that can then be fed into, our aerobic respiration processes, for example. Collagenase is an enzyme that breaks down collagen. And there's a whole bunch of different kinds of enzymes that we could talk about that break bonds and all by adding in water. So hydrolases hydrolyze large molecules to make them into smaller ones. So can you go the opposite way? So if you are building molecules and water comes out as a product, then those are ligases. And we've already talked about one ligase, and that's DNA ligase, right? So hydrolases and ligases are opposites of one another. And so if you can remember that DNA ligase joins things together to make them bigger, then remember that hydrolases are the opposite. So they're going to break things apart to make them smaller parts. Okay. We've got to break water to do that. Alrighty, what is the biggest main purpose of glycolysis in aerobic respiration? What is our biggest outcome? What are we really wanting to get out of glycolysis if we're going through aerobic respiration? We want pyruvate. Some of us are thinking, I'm talking about glycolysis, not the whole entire aerobic respiration process, which is glycolysis, the intermediate step, Krebs, and the electron transport chain. The whole goal of all of that is to make ATP. But in aerobic respiration, glycolysis is just the first step. And the whole point of glycolysis and aerobic respiration is just to make pyruvate, so that in the intermediate step, you can convert pyruvate into acetyl-CoA, so that in Krebs, you can steal the electrons off of it, so that in the electron transport chain, you can just shuttle those electrons through, build up your proton motive force, and then feed protons through the ATP synthase. So the real purpose of glycolysis is to make pyruvate. If we were going through fermentation, then you would only do glycolysis. Then your real goal of glycolysis would be to make ATP if you were fermenting, right? So. What is the biggest main purpose of the Krebs cycle? Just the Krebs cycle. What is the main goal of the Krebs cycle? All right, so good. So this is making NADH and FADH2. I was going to write on it, but you guys don't need me to because most of us got this. This is our electrons, right? This is our electron source. So NAD and FAD are electron carriers, and they're carrying electrons in the form of H's here. The H's come with the electrons. All right, next. What is the biggest purpose of the electron transport system or the electron transport chain, whatever you want to call it? the electron transport chain. All right, so this one is where we're making our ATP. This is where our ATP. And just a reminder here, how much carbon dioxide do we make in the electron transport chain? Anybody remember? How much carbon dioxide is made in the electron transport chain? None. Where do we make carbon dioxide? Krebs and the intermediate step, that's it. We do not make carbon dioxide in the electron transport chain. So just a reminder of that. I need to review that before the exam comes. All right, potential energy of the proton motive force. What is it for? We have a little image of a dam. I wonder why that would be, because it's very similar to how dams function. So what's the purpose of the proton motive force? Right, so it produces ATP. So again, what happens is just like in this dam situation, we are building up all of these H pluses outside. They can only come in through ATP synthase. And when they do, it powers the ATP synthase to make ATP. Okay, what is oxidative phosphorylation? Right, so we know that it involves electrons being moved because it's called oxidative, right? So oxidative always, that's always going to be referring back to redox reactions and exchanging of electrons. So in this case, electrons are going to be lost from one thing, which will be our donors, and then moved to another thing, which will be the acceptors. In the case of the electron transport chain, that's our cytochromes. So we're moving them off of NADH and FADH2, taking the electrons and moving them onto our cytochromes and shuttling them through the electron transport chain. The movement of those electrons from donor to acceptor is what drives creating the proton motive force. And that allows us to phosphorylate ADP into ATP. So that whole process of the electron transport chain is really oxidative phosphorylation. And that's how we're making ATP from the electron transport chain. Good. All right, so where do eukaryotic cells make their ATP in the electron transport chain? Where is the electron transport chain in a eukaryotic cell? Where does the electron transport chain happen? So this happens on the folds of the inner membrane of the mitochondria. This is called the mitochondrial cristae. True or false? Bacteria don't have an electron transport chain. So this is false. They do have an electron transport chain. And where would it be found in bacteria? On their inner membrane, on their inner membrane. Remember, we had a quiz question about this. It said to compare and contrast the locations of the electron transport chain in eukaryotes versus bacteria. All right, what do facultative anaerobes do to make energy if plenty of oxygen is available? Facultative anaerobes, if they have plenty of oxygen, how are they going to make energy? All right, so yes, they will do aerobic respiration. So facultative anaerobes can switch between fermentation and aerobic respiration. If they have enough oxygen, they will go through aerobic respiration. If they don't have enough oxygen, then they will go through fermentation. And so these are our fermenters. There aren't separate fermenters that only ferment. That's not a thing. If you're a fermenter, you're a facultative anaerobe. Good. In bacteria, where is the electron transport chain? All right, so on their inner membrane, also known as their cell membrane or plasma membrane. Good job, guys. All right, true or false? Facultative anaerobes can use oxygen in metabolism. Similar question to what we just asked, just approaching it from a different angle. True. Beautiful. So if they have it, they will use it. Beautiful. All right, true or false? Fermentation releases carbon dioxide that is made during the Krebs cycle as a waste product. So this is false, because if you're going through aerobic respiration, then you will make carbon dioxide during Krebs or your intermediate step, yes. But fermentation is only glycolysis. So if you're thinking here, like when you ferment, you can make carbon dioxide though. Yeah, in glycolysis, but it's like part of getting rid of pyruvate. They convert pyruvate to carbon dioxide, lactic acid, ethanol, and other things like that. Okay, so it's like an extra step added on to glycolysis to get rid of pyruvate and the electrons that they make. So they don't make carbon dioxide in the Krebs cycle because they literally don't go through Krebs. Okay. Good. All right, next. Select all that apply. Don't you love these? So which of the following would plants undergo? Think of cellular respiration as a representation of aerobic respiration. So they will do both. Obviously, they do photosynthesis, but they also do aerobic respiration. They have mitochondria too. Mitochondria, the only function of them is aerobic respiration. Plants do both. All right, I feel like we covered a lot of that stuff. I'm trying to think. All right, moving on to chapter 13, true or false? Your biome stops changing once you're an adult. So this is false. I'm glad that we generally agree on this. So definitely there's a lot of times when your biome can change. Think of like your, like I will say this because we're going to talk about this in Unit 4 a lot, but think of like the vaginal biome, for example. When we go through puberty, obviously it changes, but when you get new sexual partners, your biome is going to change. And when you go through, women go through their monthly cycle, their hormonal changes change their biome as a result of pH shifts from hormones. So your biome changes regularly, all the time. And again, if you get a new person living in your house, your biome can change just from that person living in your house and being exposed to their biome. So it's constantly changing. You're never done with it changing. All right. Select all that apply. Which of the following would be considered virulence factors? Maybe you're looking at some of these terms and you're not sure what they mean. I'll talk about them a little bit. See what we know first. All right. So exotoxins are toxins that are secreted by pathogens to cause damage to the host. So this could be things like collagenase, hyaluronidase, or toxins that are made like botulinum toxin or tetanus toxin or something like that. They cause damage to the host and they're secreted outside of the bacteria or pathogen. Those are exotoxins. And virulence factors, just before I get too far ahead of myself, a virulence factor is any trait that can help the organism cause infection and disease. So would an exotoxin be able to help with that? Yes. Would flagella and motility be able to help with that? Yes. Would Fimbriae are structures on the outside of bacteria that help with attachment to surfaces. Okay. We learned about that in chapter 4. So would that be helpful for them to cause infection? Yes, absolutely. And then capsules, obviously, for attachment to surfaces, for creating biofilms, and for evading phagocytosis, beneficial for virulence. So all of these. All right. Which of the following would not be considered a sign, but instead a symptom? One of these would be considered a symptom. So signs are things that you can measure. Symptoms are things that are reported but can't be measured. So nausea is not something we can measure, but we can measure fever. We can measure leukopenia, which is low white blood cell count. We can measure lymphadenopathy, which is swollen lymph nodes. And if you ever get on my exams and you're like, I don't know what lymphadenopathy means, you can ask me. Worst case scenario would be if it's a term that you have to know and I tell you you have to know it. But I'll usually be helping you out somehow. But lymphadenopathy would be a fair thing to ask about. Swollen lymph nodes. Okay. Right, you've been 100% all day, but now your throat is starting to hurt and you think you might have a fever. What phase of infection would that be considered? Sounds like initial signs and symptoms are showing up. Alrighty, so this would be the prodrome or prodromal phase. This is the first time you start seeing signs or symptoms. The acute phase, also known as the infection phase, that's essentially the full-blown illness. At its worst. Convalescence is when you get better. And incubation is the period of between whenever you got the pathogen inside of you and when you start showing your signs and symptoms. So it's the before times. When the thing got inside of you, but you haven't gotten sick yet. It's the incubation period. Any questions about that? All right, next. Former President FDR was bound to a wheelchair as a result of polio infection, even after he cleared the infection. What is the term for this situation? So we're confirming that he no longer has the pathogen, but he's still got effects of having had it in the past. The term for this is sequelae. Good job. So it's the sequel of having the illness. It's a continuation of effects of the illness. For example, I'm still getting like wicked muscle cramps from having COVID. And I don't know what that's about. All right, so that's chapter 13's review. I'm gonna move on, I think. Chapter 14, which term applies to the frequency of deaths in an outbreak of a disease? The frequency of the deaths. We'll talk about all these terms and what they mean. We'll do a little mini review of them. See what you can tell me, if you can recall. The number of deaths. We've got an outbreak going on. People are sick with COVID. If we want to look at the number of deaths associated with it, and that would be which term? Mortality. Good. So a lot of people are pretty solid on mortality. That's usually not a big issue for people, but we get confused with prevalence and incidence and morbidity. So we'll just briefly touch on those. Prevalence is the total number of cases that are in a population, total number of active cases. Incidence is the same thing as morbidity for our purposes. And this refers to the number of new cases in X time period. So how many new cases have we had this week, for example? So if we had a COVID outbreak going on. There's two different numbers. Well, there's three different numbers we'll be looking at because mortality, right? Mortality is deaths. All right, so if we have an outbreak of COVID going on, we might say there are currently 100 people with the disease, but there were 50 new cases this week and 10 people died. We've hit all of those three. We can see the difference between them, right? Okay. So we're talking about the number of cases this week. That's incidence or morbidity. We're talking about the total number of active cases. That might be a different number than the number of cases we had this week. New cases, right? Okay. So that's epidemiology in terms. Love it. Moving on. Which aspect of testing is determined by how few people test falsely positive? This will be specificity. If I ask you which aspect of testing is determined by how few people test falsely negative, that would be sensitivity. If we're talking about false positives, that's specificity. And the exam question will not say the words false positive or false negative. It's going to give you a scenario, and you're going to have to infer what a false positive and a false negative is. Does anybody need me to cover that in many more detail before we move on? Okay. So remember, false negative means that you tested negative when you were actually positive. False positive means you tested positive, but you were actually negative. So you take a drug test today and you test positive for methamphetamine when you took Sudafed, not meth. That would be a false positive. Okay. It's not a very sensitive test. We good? Good. All right, we're getting there. We're almost there. What type of epidemiological study involves watching uninfected people over time to see if they get infected? All right, so this is the cohort study. The reason we know it's the cohort study is because it tracks it over time. And I'm glad that nobody chose Hill's criteria. Hill's criteria is covered in the chapter, but you don't need to know it for my exam. You will talk about it if you go into the nursing program, though. And it has to deal with tracking epidemics, basically outbreaks. Really important when you start having outbreaks in hospitals, which you will all the time, I'm telling you. So even if you're not even thinking about what do you mean you have outbreaks in hospitals? Even C. diff spreading from room to room, that's an outbreak. So stuff like that, you can track outbreaks like that using Hill's criteria and larger populations as well. Anyways, cohort study, looking at whether or not somebody gets the disease over time. Usually we compare a risk group and a non-risk group, smokers and non-smokers, and then following them over time, which group ends up with more lung cancer. That would be a cohort study. Cross-sectional is going to be instantaneous. Oh, we have an outbreak of COVID. Just take a whole bunch of people, test them for it, and see if they had any exposure factors. Well, we have an outbreak of COVID, and we're going to gradually gather people who have it and gradually gather people who don't, and then compare the differences. That would be a case control study, comparing cases with control groups, but not waiting to see if they're sick, letting the people be sick, and then sorting them into groups. That makes sense? Okay. All right. 66. Rules for hand washing, changing gowns, cleaning surfaces, and similar practices. Who is in control of that at a hospital? This is covered in chapter 14. All right, so this is the infection control officer. Pretty straightforward. I mean, your charge nurses and the hospital board and the treating doctor might be a little involved in that stuff, but the person that made the rules for that stuff, that's going to be the infection control officer, and everybody else is just following their recommendations. All right, what is the term for the number of people an infected person will likely infect while they're sick with an infection? These are all terms that we do have to know for chapter 14. And we'll kind of touch briefly on those when I'm done with the unit 2 review. All right, so R naught is the number of people you're like. So this is the measure basically of how contagious a pathogen is. CFR is case fatality rate. This is if you get the disease, how likely it is you'll die if we don't treat you. Okay, so that's case fatality rate. HAI is hospital associated infections. I'm telling you, if you're going to go work in a hospital, you will get to know these very, very well. We're talking about, catheter associated infections, respiratory equipment associated infections, C. diff and things like that spreading within hospitals. All right, and morbidity, again, is a measure of new cases in a certain time period. All right, next question. What is herd immunity? Isn't really so much a definition as a representation. All right, so blue is our best answer here. In herd immunity, we have enough people vaccinated that the pathogen isn't going to spread from person to person and therefore can't spread to people who are not vaccinated. It doesn't mean that the whole population is immune. And it doesn't mean necessarily that it provides protection only to those who are already immune. But the fact that we have people who are immune, the pathogen can't spread in the population, and therefore the people who aren't immune can't get infected. This can be conferred by either vaccination or by natural exposure, like we have with the chickenpox. Not as much now, but when I was a kid, the goal was to get you the chickenpox when you were a kid so that we could create herd immunity so other kids wouldn't get it later on in life. Same idea as with vaccinations. All right, that is it for our Unit 2 review. Does anybody have any questions about anything? Did I not cover something you want me to cover? Okay, beautiful. So I will record this. This will be posted for you guys. If this were the exam today, here's our podium really quick. Now you guys know Jolette, Megan, and Noah are the ones to come for, all right? They're the ones. All right, we did great on this. Let's see. Unit 2. That was a 43 average. Just FYI, we got plenty of time to study on this. I know we weren't going to take a test today, but FYI, okay? So good job today, guys, and I'll see you on Thursday for Lab 12. It should be straightforward. Right, good job.